Hi friends, welcome to classes by Lab Doctor. I'm Dr. Divya and in today's class let us study about the uric acid. So in the kidney function test, one of the important parameter is uric acid. So what is this uric acid? Uric acid is a waste product. And it is a waste product of what? It's a waste product of purine metabolism. And where do we get these purines? We either get it from the foods or the drinks or from our own cells. When our own cells die or when the cells there is a turnover of the cells, we get this purines. So ये जो uric acid होता है that dissolves in the blood and जो whatever is there it is excreted by the kidneys. But any condition where there is an overproduction of purines or under excretion of purines, the time we get hyperuricemia. Hyperuricemia is very common. 1 in 5 people will have this hyperuricemia. What are the complications if we have increased uric acid level in the body? So ये मत सोचना कि अगर uric acid level ज़्यादा है only you will get gout. No, this increased uric acid can damage the heart, it can damage the brain, it can damage your liver, it can damage the insulin resistance. All this along with the involvement of bones, joints, tendons, ligaments, kidneys, etc. So I've told you that there are various factors which are associated with this hyperuricemia or increased uric acid, like hypertension, dyslipidemia, obesity, metabolic syndrome, medications, kidney disease, diabetes, the gender, aging, lifestyle. All these are interrelated and all of these can contribute to increased uric acid in your blood. So first let us study about the uric acid homeostasis. So purine comes mainly from the diet which we consume. Okay, suppose if a person is having a meat, organ meat, alcoholic beverages or sugary drinks, beer, etc. ये purine इनमें ज़्यादा होता है and the content of purine will be more. And purine is also formed by the cellular metabolism of nucleic acid. ये purine and pyrimidine क्या है? ये nitrogenous bases है जो हमारे DNA and RNA में होते हैं. तो जब भी cell turnover होगा, when the cells will be broken down etc., the amount of purine will become more. So that will contribute to the uric acid pool which is formed by the liver. So in this liver only the purines को हम uric acid, the waste product, convert करते हैं. So this uric acid when it is formed, 2/3 kidneys will excrete it, and this uric acid is freely filtered in the glomerulus and 90% is reabsorbed in the proximal tubules, and the remaining 1/3 what is there, that is excreted in by the GI tract. So now I've told you कि ये uric acid is the end product of purine metabolism. So what are these purines and pyrimidines? ये purines and pyrimidines are nothing but the nitrogenous bases that are the building blocks of RNA. So purines जो है ये एक नाइट्रोजिनस बेस है जो हमको डीएनए एंड आरएनए बनाने में चाहिए. सो प्यूरिन्स कौन से हैं? एडिनिन एंड ग्वानिन, ये हमारे प्यूरिन्स है. इनके ब्रेकडाउन प्रोडक्ट सी है, that is uric acid. So the waste product of this purine metabolism is uric acid, and purine and pyrimidine are required for formation of DNA and RNA. So whenever there is a cell breakdown, obviously the uric acid is going to increase. So जिस भी condition में uric acid बढ़ेगा, that condition the cell turnover will be more, like hemolysis, rhabdomyolysis, tumor lysis. When the cell will lyse, there will be breaking of DNA, RNA, and the waste products will be more. So the increased production of uric acid can be because of the increased cell turnover, like all these conditions: rhabdomyolysis, hemolysis, tumor lysis syndrome, chemotherapeutic agents, etc., which can lyse the cell and increase the uric acid level in the blood. So how do we get these purines? How are these purines synthesized? अभी ये purines जो है कैसे बनते हैं? One is de novo synthesis, newly formed purines, and second one is salvage pathway, means whatever is formed we are reutilizing it, second hand जैसे हम यूज करते हैं वैसे ही ये de novo pathway, एक salvage pathway रहता है. So purine synthesis it will always start with this phosphoribosyl pyrophosphate, and this PRPP will form the IMP, which will give rise to AMP and GMP. So this AMP and GMP are important because AMP and GMP से ही क्या होता है, ATP आता है, यहाँ से GTP आता है, जो we use it again for the production of DNA and RNA. So ये देखिए PRPP it forms IMP, IMP forms GMP as well as AMP. So this AMP is adenosine. Okay, so this is adenosine. This will later form the ATP, which we use it for again for the production of DNA and RNA. Similarly, the GMP, this will form guanosine. This is again a purine guanosine, and then later this is utilized again for production of GTP and RNA and DNA. So this purines are formed here. So ये जो purines होंगे, this is adenine and guanine. तो ये purines form हो गए. So now when the purines are formed, they will be utilized, and what after utilization whatever is left we have to degrade it. इसको हमको degrade करना पड़ेगा. तो ये guanine, ये adenine degrade कैसे होगा? ये hypoxanthine, it will become hypoxanthine, xanthine, and then uric acid. So the purines are formed here. They are utilized to form DNA, RNA, and the remaining after that they are degraded to form the uric acid. And during this process we need all these enzymes, xanthine, oxidase, etc. So this is the nucleic acids, that is it forms the purines. The purine will be degraded to hypoxanthines, hypoxanthine to xanthine, xanthine to uric acid, and uric acid is excreted. So this whatever uric acid, whenever it is formed, this waste product is formed, there are many medicines which we use in the treatment of uric acid, which will act on these pathways to prevent the increase in uric acid. जैसे कि हम लोग ये allopurinol देते हैं, it will prevent the conversion of hypoxanthine to xanthine. Yes, and once xanthine is not formed, uric acid production is lessened. So uric acid, some drugs like sulfapyrazone will increase the excretion of uric acid in the kidney. Degradation of purines, मैंने बोला आपको ये IMP से, from the IMP you get the AMP as well as the GMP, and this AMP and GMP, AMP is adenosine and GMP is guanine. So these are the purines, isn't it? So the purines will be utilized. जब purines form होंगे, so the purines को हम किस लिए use करते हैं? To form DNA, RNA. तो ये अपना काम करेंगे. They will form DNA, RNA, etc. And then they will be broken down. These purines will be broken down or degraded. So when these purines will be broken down, they will form this hypoxanthine and xanthine, and from them the uric acid is formed. This uric acid excretion also I have told you, it is like in a normal condition, 2/3 is excreted from the kidney and 1/3 is from the gastrointestinal tract. Uric acid excretion from the kidneys as well as GIT will depend upon various factors. Excessive consumption of a fatty food, sweet food, food rich in high cholesterol, high fat, they will affect the kidney function and the reabsorption of this uric acid. So all these conditions, high fatty food, high sugary food, etc., will cause hyperuricemia because they will affect the absorption of this uric acid in the proximal tubules, and hence the level of uric acid in the blood will rise. I've told you that uric acid, it is removed 2/3 by the kidney. So around 75% of uric acid is filtered by the kidney, and around 90% of it is reabsorbed, and the remaining remaining whatever 25% is there, that is excreted by the genitourinary tract. So this is the place wherein the proximal tubule, where this one is reabsorbed. 90 to 95% of the uric acid is reabsorbed and it is excreted here by the kidney. I've told you the second way in which the purines are produced is the salvage pathway. So first था जो originally synthesis हो रहा था, so there was synthesis, actual synthesis जो हो रहा था, that was de novo, from the beginning, that was de novo. Now this is salvage pathway. Salvage pathway, what happens? Whatever adenine and guanine are there which were supposed to be excreted, we are reusing it. Reuse is called as salvage. So यहाँ पे हमने बोला कि IMP generate हुआ, यहाँ पे GMP, AMP हुआ, and then from guanine, adenine, they were utilized to form DNA, RNA, and the remaining were excreted. But if we reutilize, instead of putting this guanine, तो उसको reutilize करके हम जब ऐसे हम reutilize करेंगे तो ये यूरिक एसिड का जो पूल है वो कम होता जाएगा. अगर ये सैल्वेज पाथवे ठीक से काम नहीं करता है, सो दिस ग्वानिन एडेनिन जो भी है, दे विल नॉट बी रीयूटिलाइज्ड, दे विल बी producing more and more of waste. So that time also uric acid level will rise. See, DNA and RNA, when you degrade, it forms nucleosides, the bases. We are reutilizing it, we are salvaging it. So from the salvaging also there is production of the uric acid. So I have told you that in the salvage pathway there are certain enzymes which are required. For example, HGPRT. This HGPRT helps in the utilization of this guanine, reutilization of this guanine. So agar, like in case of Nish-Nihan syndrome, this HGPRT hormone will be deficient, so the patient will not be reutilize this guanine and there will be increased level of uric acid. Whatever guanine is there, वो waste product ही form हो जाएगा, so uric acid level only will be more. So the such hormonal deficiencies can give rise to certain metabolic conditions like Lish-Nihan syndrome. So I have told you that this is a de novo synthesis wherein we form the IMP, inosine, and then this IMP forms this GMP and AMP, and this is a salvage pathway where we are reutilizing this adenine and guanine and we are using it again to form the purines, whereas both the end product of both of these pathways is uric acid. So this is the pathway where you will see that PRP formed IMP, IMP formed GMP and AMP, which were utilized to form the DNA, RNA, and also generated ATP and the GTP, and this were then broken down to form hypoxanthine, xanthine, and the uric acid. Sometimes they can be salvaged. Instead of all becoming the waste, they can be salvaged and reutilized. That can also lead to the formation of this uric acid, that is a waste product. So this is the Lish-Nihan syndrome where the child, the boy, usually has hypotonia, dystonia, balismus, developmental delay. He will have gouty arthritis and hyperuricemia because of the deficiency of HGPRT in the metabolic pathway. So this is the ribose 5-phosphate. It will lead to the formation of inosine monophosphate. So this is a synthesis of purines. So what purines we are talking here about? Guanine and the adenosine. So from the ribose 5-phosphate, inosine monophosphate is formed. From this we get this adenosine monophosphate, AMP and GMP. This will form the inosine, this will form the guanosine, and then both of them will form the hypoxanthine, xanthine and the uric acid. So this is a waste product which is formed by the breakdown of this purines, and this uric acid 75% is excreted in the urine, the remaining is excreted by the GIT. Uric acid can directly come from the dietary nucleic acids also, like the meat, red meat, organ meat, etc. So this is about the metabolism of uric acid. So the liver, the kidney, the GI tree, all of them play a role in the production of uric acid. This becomes important because any changes in any of this pathway can lead to increased uric acid level in the blood, which in the later classes we will see the causes for hyperuricemia. So liver becomes important because this whole process of production of uric acid takes place in the liver. So any problem in the liver, uric acid can rise. Any problem in the kidney, where the kidney is not able to excrete, the uric acid will rise. So this is the pathway wherein we get the uric acid. So uric acid basically is a waste product, and this is the end product of this purine metabolism. So later classes we will see what are the causes of hyperuricemia, complications of uricemia, etc. Thank you so much for watching the video. Please like, share and subscribe the channel. Thank you.